We left off speaking about the synaptic modulation, and we talked about the facilitation and the inhibition of this. And just to review here, we said that synaptic facilitation was one neighboring neuron, okay, specifically at an axonic synapse, influences this neuron C to release more neurotransmitter, okay? And as such, it is affecting the postsynaptic cell because more neurotransmitters released, but it is not directly influencing the membrane potential change. And what that really means is it's not firing an action potential. There's no electrical energy or membrane potential change that's being communicated from this modulating neuron. All that is being communicated is the relative secretion of the neurotransmitter. And likewise for synaptic inhibition, the neuron is influencing the presynaptic neuron there to secrete less neurotransmitter, okay? So make sure you understand synaptic modulation and you're able to distinguish inhibition from facilitation. And make sure that you understand here, more importantly, that on its own, this neuron does not influence the membrane potential, okay? It does not influence the change or the fluctuation in the membrane potential. Okay, and so just to point out what type of synapse this is axonic, and we said axonic was where those two axon terminals communicated, right? And this is sort of where we see this in the body. We said it was not very common, and this is the example of an area that we see it in the body. So we can either excite or inhibit another synapse. So it can be selective in that way. Okay, it's not going to necessarily give one response because it's not necessarily receiving a stimulus and passing that on. Okay, it's modulating that specific synapse in its own right based upon its own influence of that calcium release. And then axodendritic and axosomatic are the types of synapses that the modulating synapses will influence. So basically an axoaxonic synapse influences an axodendritic or axosomatic synapse, okay? And these are non-selective, okay? And when I say non-selective, it means whatever type of signal or communication is coming in from that presynaptic neuron, that's what it's going to fire, okay? If it's getting graded potentials that are IPSPs, it cannot determine between those two things, okay? All right, so let's sort of summarize here everything we've spoken about with regard to the synapse. In terms of the anatomy, we talked about the soma, the axon hillock, the axon itself, and the axon terminal. We also spoke about different types of relationships, so convergence and divergence, right? This idea that neurons don't synapse one-to-one the way we've kind of been discussing them. They actually have many different branching patterns and are highly branched in that way, okay? And so on the presynaptic neuron, we spoke about what was happening here. We had an action potential being propagated down the length of this neuron. This triggered the opening up of voltage-gated calcium channels, and that would eventually release some sort of neurotransmitter into the synaptic cleft. Okay, once that neurotransmitter was released into the synaptic cleft, it would specifically bind onto a receptor on the postsynaptic cell. We spoke about a lot of options for receptors, but two broad categories. Can anyone remember the two types of receptors? So we spoke about two on the postsynaptic cell. We got an enzyme on there. But in terms of the receptor, what type of receptors did we talk about? So we talked about ionotropic receptors and metabotropic receptors, okay? Ionotropic and metabotropic, right? Make sure that you're tying all this information together, right? Students have a tendency to sort of compartmentalize this, okay? The receptors that we talked about are on the postsynaptic side. Make sure you're making those connections. All right, so on this postsynaptic cell, we have an ionotropic receptor or a metabotropic receptor. If it were a metabotropic receptor, we would bring about what type of response, fast or slow? Slow, right? Slow response. The metabotropic receptor, we said, had several different players, several different messengers involved. This is what we call the signal transduction cascade or the signal transduction pathway. It involved a very important player. What was the most important player in the metabotropic pathway. I'm hearing it in the back. G proteins, perfect, G proteins.